Last week, I've just been throwing all this immunology at you. And it's pretty clear everybody's really scared, afraid of how much they have to know. And so what I'm going to try to do this morning is kind of integrate a lot of what we've talked about. Not everything, but a lot of it. So you can see how this works when you encounter an antigen and how the immune system begins to handle that antigen all the way through adaptive immune response. Okay? So this is the time for you to ask questions, and I'm going to ask questions, and we're just going to get a gauge of how everybody's feeling and what you know. And as I've mentioned before, the key thing for you for studying is the study objectives. If you can look at those study objectives and say, I know that, I know that, I know that, that's where almost all the questions are going to be focused on, because there is a lot of material, but some of it has to be brought in here so you can understand the context and how everything works and stuff. So I know there's minutia of one and probably. So the one thing that you really need to understand about immunology, even though we kind of subdivide it into innate and adaptive immunity, is that the system works together. And it works best together, and you can tell that because if you're missing a component in any one of the systems, you're going to have some problems, some immunodeficiency that's going to leave you predisposed to some sort of an illness, whether it's bacterial or viral, or you end up with some sort of weird syndrome or something like that, autoimmune disease, all kinds of different possibilities. So the system integrates all the way across the board. The adaptive immune system can't do its job unless the innate immune system is helping, generating cytokines and camps and complement fragments and all kinds of things, because they all have receptors for that stuff too. And that helps to drive their particular direction for an adaptive immune response. Okay? So, say you're in lab and you've got a nice petri dish full of some bacteria, and your lab mate takes a nice loop and just kind of scoops a whole bunch up and just goes, flicks it at you. Gets in your eye, gets in your mouth, gets in your face, gets in the cut because you just shaved today. So the question is, are you going to die? Yeah. So the answer is yes, you're going to die. Okay. So now yes.
Do you mind if we turn up the volume just a hair? Oh, okay. Sorry. Thank you.
Yeah, I get this.
Well, not that much.
How about this? Is that better?
Yeah.
How's that? Better?
Maybe you move the mic up higher on your shirt.
Okay, better? Good. All right, so now we've got these bacteria scattered all over the place. The stuff that's in your mouth and in your eye, that's mucosal immunity, right? We learned a little bit about that yesterday. We kind of talked about it at various times and stuff like that. So the mucosal immune system is going to come into play to try to handle the microorganisms that have gotten onto those surfaces. Things that have gotten under the skin, innate immunity is going to be an important factor in controlling that. Things that have gotten into the cut, well, now it's a little bit deeper in. And maybe that bacteria is found a niche to start to multiply and cause a localized infection. And so what's going to come into play to help protect you there? So the first part that's going to be involved here is innate immunity. And what do we know in general about innate immunity? One, that it has no memory. So the next time this friend of yours does this, you're not going to respond any better to the pathogen any faster through innate immune mechanisms, right? It doesn't remember that pathogen. It just recognizes what on that pathogen? The PAMPs, right? So the innate immune system recognizes PAMPs, pattern associated molecular patterns. So those are things that are found in microorganisms that are not found in humans. So things like LPS, bacterial peptidoglycan, cell wall material, different types of RNA and DNA structures that are found in viruses, but not in your own cells, those kinds of things. So that allows the innate immune system to recognize hundreds of thousands of different types of pathogens with just a handful of receptors. Okay? And there's no rearrangement of genes in those. Those are germline encoded and they stay that way. And so what's the main family of molecules that recognize the PAMs? That's right, the toll-like receptors, TLRs. Okay. So those are very critical for that. One of the advantages of the innate immune system is that it acts immediately. So as soon as that pathogen gets into the eye or the mouth or into the cut or something like that, you're going to start stimulating a variety of different mechanisms. They're going to try right away to eliminate that pathogen. And it may do that so efficiently that maybe there's not even really a good adaptive immune response occurring, because maybe the pathogen numbers are small and they were easily taken care of, and that's the end of that. Okay, so that's one possibility. So no memory, acts immediately, but unlike the adaptive immune response, it's not so specific, right? It's not recognizing little epitopes like an antibody does on the surface of a pathogen. It's not recognizing peptides like a T-cell receptor does, okay? So it has some pros and some cons. So let's talk about the ones that have gotten into any of these surfaces. So the first line of defense in innate immunity is going to be things like skin, or for those things that have gotten into the eye or to the mouth. Then we're talking about things like saliva, mucus. Okay, let's spell that wrong. Okay, so those are the first barriers, physical barriers, to prevent pathogens from going any further. So that's one part of the innate immune response. It provides a barrier to prevent pathogens from getting deeper into the tissue so that they can cause an infection, right? So physical barriers. Okay. And the skin, it turns out, has molecules like defensins, natural antibiotics. It has just a slightly lower pH. So microorganisms that have a strict pH requirement, they may not be so happy there, so some might not be able to survive in that environment. Okay? And there are also some lipids, natural lipids there, that also have some antimicrobial activity. So you've got the physical barrier, and you've got some of these physiological mechanisms that help eliminate that. In these tissues, you have things like lysozyme, okay, which is an enzyme that degrades peptidoglycan. So bacteria that have thick cell wall, they're going to be hampered in their ability to survive in that environment. Okay. You also have molecules like defensins, so natural antibiotics that can help. And although not part of the innate immune system, you also have here antibody. And what's the major antibody that's found in mucosal surfaces? IgA. Good. Come back to that. Okay. If you got enough in your mouth and you ended up swallowing some of it down to your flora, it's going to hit the stomach. And not a lot of bacteria survive in the stomach because the pH is really low, right? pH 2 to 3. And we've got digestive enzymes. So if you put 10 to the 2 or something like that bacteria in, only about 10 to the 10th, only about 10 to the 2 are going to come out of the stomach. So it really whacks the number down in that environment. Okay? So pretty tough. If it makes it further down, like into the small intestine, then you've got things like bile salts. And the pH is high now. So you've gone from low pH to high pH. Not a lot of organisms can survive that kind of a transition. So it's going to be pretty tough on them. And then if it makes it down even further into the large intestine, remember yesterday, Dr. Lorenz was talking about mucosal immunology. That's where all the bacteria are. You've got a ton of bacteria in the large intestine. And so then you've got competition with the microbiome, the natural microbiome that you have. And those guys are very protective of their turf. They're happy there. They get all the food they need, they stay warm and all of that, so they're not going to just let anybody else come in and try to take over if they can help it. Okay? So lots of mechanisms to make it very difficult for this pathogen that's got into your eye or you get swallowed or something like that, that has started to try to cause an infection. So what about the one where it made it into the cut? Can I have a cut broken into the skin here? Got the bacteria starting to replicate. Maybe they're starting to get inside underneath the epidermis into the dermis. So what are they going to encounter in the skin? What type of cells are going to be immune cells that are going to be present there? So macrophages are everywhere. Every tissue. Brain, liver, lung, skin, anything. Macrophages are everywhere, right? So you're going to have macrophages. And you're going to have skin mast cells. Okay. And what other really important cell type do you find in almost all different tissues? Cellular? No? Antigen presenting cells. Right? So dendritic cells. And the key one in the skin is a cell that has the name a Langerhans cell. Named after the guy who figured out that that's what they were. These are the same kind of thing. Right? So now you've got the potential for all kinds of responses to help eliminate that pathogen, right? So when that bacteria encounters the macrophage has TLRs on its surface. That'll make it angry. You're going to get some local production of different cytokines. Okay, and in the case of a bacterial infection, those are going to be pro-inflammatory cytokines, things like TNF-alpha, IL-1. Okay. There are others that worry about that. So the macrophages are going to start getting angry in that environment. There's also in interstitial fluids, and there's also going to be blood vessels running by here. Okay. You're also going to have another important part of the innate immune system, complement. Okay. So when complement encounters that pathogen, it can be activated and it can release all kinds of different fragments that do a lot of different things, right? So chemoattraction through 3A and 5A. And what's it going to chemoattract? It's going to chemoattract other macrophages in the surrounding area. And in the blood vessels, you're going to have those lovely little neutrophils, also called PMNs. So they're going to get attracted out to the site of infection. Okay. Complement can also directly kill those pathogens through what structure? MAC, membrane attack complex. Okay. So it can directly kill. It can chemo-attract phagocytic cells. Okay. And then the other important thing the complement can do is it can opsonize, it can target the pathogen for clearance by phagocytic cells, right? So it does that through C3b and C4b. So this is MAC. This is things like C5a. And then it can also opsonize. That's through things like C3b. So now that bacteria has on its surface C3b, fragments derived from that, and macrophages have receptors for that, and it makes it easy for that macrophage then to engulf that pathogen, phagocytosis, right? And that's going to then kill that pathogen. What other molecule is important in opsonization? For phagocytosis? Fc receptors. Antibody? Right? So there's antibody and complement-mediated phagocytosis. So maybe you've seen this pathogen before, and you already have some antibodies floating around. So you can also have antibodies bind to the surface of this pathogen. And that would be really nice if you had both of those, because then the pathogen is really well targeted. It's going to be phagocytized by macrophages. And the neutrophils are going to come across, and they can also phagocytize. And what else do neutrophils use to kill cells besides phagocytosis? They have those structures called nets. You can throw those nets out, kill a lot of pathogens in a broad area around them, outside the cell. Okay. And so we spent some time talking about how it is that neutrophils can actually get from the blood into the surrounding tissue. So that's when these mechanisms come into play, right? We've got an infection now in the surrounding tissue. We need to get these neutrophils to stop rolling. The first step that they're always doing in these small vessels, right? And rolling is selectins and carbohydrate ligands. Right? So then you're going to get the signaling event that occurs, activation that occurs, and that's going to lead to a conformational change in integrin molecules on the neutrophil so that it can firmly adhere. So here we're talking about integrins and their ligands, some of the cell adhesions, the CAM molecules like ICAM1 that lead to that firm adhesion step where the neutrophil flattens out and sticks to the endothelial cell layer and then transmigrates into the surrounding tissues. Right? So this does 4 steps. That's what you need to remember for that lecture, those 4 steps and the molecules that are involved in that. Selectins and carbohydrates, integrins and ICAMs. And for transmigration, there's the JAMs and the PCAM and stuff like that. So that's important, so you can get these guys out of the blood vessel. So if you're missing some of those guys, neutrophils can't get out, your ability to phagocytize and kill is going to be limited under those circumstances. So what about the mast cells? Mast cells probably contribute more than we realize to this, and they can be activated by lots of different things. So they can get activated by cytokines, get activated by PAMs, and they can get activated by complement molecules like C5. And even the physical stress of the cut can activate macrophages. They're very sensitive cells, and so because their job is to try to protect, really. So they can do all of that. And then they're going to start to degranulate, and they're going to release all kinds of different things when they do that, right? Those granules contain a ton of different mediators. The most important one is going to be histamine. There are other vasoactive amines that they produce. And that's going to be important because it's going to cause the endothelial cell layer to be a little bit leaky. So it's going to make it easier for cells like neutrophils and macrophages that might be in the area to crawl in here. It's going to allow serum components like antibodies and other enzymes, other immune-mediated proteins to get from the blood into the surrounding tissue. And then that's going to lead to edema, swelling, and redness, and all of that that's associated with an infection, which is a normal, important mechanism for killing. So inflammation doesn't mean something's really bad. It just means the immune system is doing what it's supposed to do in that environment. Okay? So lots of players to this point. Any questions? Not saying a lot, I'm a little concerned. Are you understanding what I'm talking about? No? It seems of no. This is why you ask questions. Okay? All right, so we've got a lot of stuff up here. It's amazing, actually, that bacteria can survive at all in this kind of an environment. What if this was a viral infection? So if this was a viral infection, then any cell in the area could be infected, right? And the virus is going to bind to some sort of a receptor that it's specific for. It can be a lot of different things. Viruses have found ways to grab onto almost anything to get inside a cell, and they're going to start replicating and releasing virus, right? So if that was the case, complement could play a role in that to help eliminate that infection. Macrophages can get involved, potentially phagocytizing the viral particles. In terms of cytokine responses, right, because viruses are going to induce a different subset of toll-like receptors. And when you get a viral infection, what is the classic signature cytokines that are produced in a viral infection? Interferons. So if this was a virus, you're looking at interferons. And there are a couple of types of interferons, type 1 and type 2. Every cell in your body makes type 1 and type 1 interferons, so alpha and beta. And that's important so that you can limit the ability of the virus to replicate in the cell. And basically what that does is it reduces expression of the MHC class 1 molecules. And it also tells the cell you're going to have to die in order to limit the spread of this virus. So it induces mechanisms that chew up RNA, block protein synthesis, and the cell undergoes apoptosis and dies to limit the viral spread. Okay. Gamma interferons type 2, and that contributes to that process also, but it's also a very good activator of macrophages to help them become phagocytic and so forth. But it also is important in the adaptive immune response to the viruses because it helps push that process along. Okay, let me see if I've forgotten anything about the cover. Okay, all right, so now let's move a little bit towards the adaptive immune response. So, where are they? Here they are. Dendritic cells, Langerhans cells. These guys are going to help be important in kicking off the adaptive immune response. So what they're going to do is they're just going to bind antigen that's come in, right? And they're going to traffic this through what structures, right? So all these structures all over your whole body that are designed to drain
lymphatic system,
lymph, right? So lymphatics. Okay. Okay. So that's a secondary circulatory system. It's not connected to the standard vasculature, so it doesn't have a pump. It's got all those valves that keep it from backflowing, and movement of lymph is dependent on your own movement and muscular contraction. Okay? So that pumps that. So that's a mechanism that is important just for keeping your interstitial fluid levels at an appropriate balance, because fluid in the interstitial tissues will move through the lymph and be dumped back into circulation through the thoracic duct. But it also is a way to get antigens to what structure? Lymphatics go to the
Lymph node.
lymph nodes. Okay. And you have nodes scattered throughout the entire body, so wherever you might get a cut, there's going to be some regional draining lymph node that will pick up that antigen and be able to start an immune response. And in fact, within the last year, they've actually figured out that there's a drainage mechanism for the brain which doesn't have any lymphatics or lymph nodes, but manages to get antigens out of the brain and into the regional lymph nodes so that you can have a response there. Nobody thought that was true for a long time. Okay? So lymph nodes are an example of what? Secondary lymphoid tissue. It's another example of secondary lymphoid tissue. Spleen. Where is that? I'm not hearing it.
Payer's patch.
Peyer's patches, exactly. Okay. Tonsils and adenoids. So what's a primary lymphoid tissue? Thymus and bone. Okay. Primary. Thymus. So what's the really important cell sitting in the bone marrow that's critical for immune responses? Hematopoietic stem cells. So they self-renew, and then they also undergo differentiation in a very controlled fashion to generate lymphoid type cells, right? T and B cells, and myeloid cells. So all the subsets of neutrophils and macrophages and basophils and eosinophils and red cells and all of that kind of stuff, dendritic cells, those kinds of things. So that's really important. Is this the site where T cells mature? No. So the T cell precursors go to the thymus, and that's where they mature and are released to the periphery. So I think we kind of touched on some of the functional roles of these guys. One other cell type that is produced in the marrow is the megakaryocyte. That one. And that's responsible for the generation of platelets, right? And platelets they're anuclear, but they're littered with all kinds of different molecules, and they can be tweaked by complement and all kinds of different things to release their contents that can contribute to the inflammatory process and also help in the adaptive immune response as well. Okay? So in the adaptive immune response, we're talking about humoral immunity, cell-mediated immunity. Okay. And humoral immunity is mediated by B cells, right? And antibody. Cell-mediated is T cells. And there are subsets of T cells that are critical for immune responses. So there's CD4, these are the helper T cells. CD8, which are the cytotoxic. Okay. So the CD4 cells produce lots of cytokines to help direct the adaptive immune response. And the CD8 cells are the ones that will target virally infected cells or cells with intracellular pathogens so that they can kill those guys and get rid of them. Okay? So the adaptive immune response has different features than the innate immune response. It has memory. So when you encounter a pathogen, you'll generate memory T and B cells, and they can be very long-lived, in some cases decades. People have shown they can live for very long periods of time. And that gives you a faster and a heightened response the next time you encounter that pathogen. Right? The IgG levels will be much higher the second time you encounter that pathogen compared to what they were the first time. Because now instead of maybe only having 5 or 10 cells specific for that particular antigen, now maybe you have 100, 200, 300, something like that. So just by sheer numbers, you're going to have a better chance of encountering those cells to eliminate the pathogen the next time it comes around. And those cells are already kind of in a prime state, ready to go. So they don't have to go necessarily through the full activation mechanism that T cells do when they're naive, encountering pathogen for the first time. So the system has primed itself now to work better the next time you encounter that pathogen, right? So you have memory. The other feature that's really critical for the adaptive immune response is specificity, exactly. Incredible specificity. So just change one amino acid and boom, you combine that one instead of that one. So it's really amazing the different levels of specificity. And it's been estimated that you can recognize anywhere from, and depends on the book you want to look at and people's calculations, but you can recognize all kinds of pathogens. So, how is it that you can recognize all those kind of pathogens? What is so unique about T and B cells that allows that level of specificity in the immune system? What happens in T and B cells doesn't happen in any other type of cell? Gene rearrangement, right? T and B cells undergo gene rearrangement. So they take little segments of genes and they randomly put them together. When you randomly put them together, you generate a new sequence, and that new sequence gives you a different structure, and that different structure gives you a different binding capacity for that antibody or that T cell receptor. Okay? So that's a really unique feature for the immune system and for T and B cells. No other cell does that. There's risk in that, and then you can make autoimmune responses in that scenario. That wouldn't be so good. And of course, what happens quite frequently is that you have an unproductive rearrangement. So you generate a soft codon. And you get a truncated molecule, and that happens a lot. You lose most of your T cells going through the thymus between that mechanism and selection to try to pick the ones that are not reactive to you. Same thing for B cells. You lose at least 50% of the B cells just because of that rearrangement mechanism. So the immune system is horrifically wasteful. And it requires a huge amount of energy. They're just starting to understand how the immune system deals with energy requirements and so forth. Pretty amazing. So T and B cells undergo gene rearrangements, and for T cells, you get a T cell receptor, and that T cell receptor has an alpha and a beta chain, right? That alpha and beta chain generates a receptor that is specific for what? For the T cells see. T cells see peptides, remember? So these guys only see peptides. And when you rearrange genes in a B cell, you generate antibody. So antibodies are a little bit more structurally complex. So you have what makes up antibodies? Heavy chains, light chains, right? And in addition to having light chains, you have different isotypes of antibodies, right? So it's the heavy chain that specifies the functional activity of the antibody through the FC region of the antibody, right? So antibodies look kind of like this. This is the FC region, and this is the antigen binding portion. So all B cells start out making IgM, And then depending on the pathogen and the signals that they get, they can isotype switch to other types of heavy chains. So they can then become IgA or IgG or IgE. IgD is out there, but there's not really much IgD, so you don't even need to worry about IgD until somebody figures out what it does and everything like that. So don't need to worry about that. So those are the major types. So these are the guys that are in mucosal surfaces, also in the serum, but also one of their primary functions is to mediate protection in mucosal surfaces. IgE is in blood, serum, plasma, whatever you want to call it, and also in interstitial tissues. So if we bring out the interstitial fluids, we can always find some IgG in there too. So antibodies are basically everywhere. They're in tears, all mucosal secretions, all over your body. You can get protection. IgE, not so much in serum. What do we think about for IgE? Classically, sorry, hypersensitivity, type 1 hypersensitivity for allergies. So kind of the bad side of antibodies. Antibodies. So can I erase this? And we'll try to finish up in 10 minutes or so. So now we've got our pathogen oxidized and bound to a dendritic cell, and it's dragging it through the lymphatics. And independent of antigen, we've already made a huge repertoire, millions of T cells and B cells, right? So that gene rearrangement event that occurs to give you all those different T cell receptors and all those different antibody specificities, that occurs independent of the presence of antigen. That's occurring in the bone marrow or in the thymus, so that those cells haven't seen antigen yet. So the immune system is basically preparing itself for things it's never encountered, in hopes that it has a broad enough specificity to be able to handle whatever it does happen. Okay? So we've made all these guys, we're all sitting around and waiting to do that. So we're going to reverse the direction here. Okay? So that dendritic cell that has brought that antigen in, and antigens can just move through the lymphatics by themselves. They can get taken up there and get pushed along. So you can have completely intact antigen flowing along the lymphatics, and you can have dendritic cells that have grabbed those guys and carried them into the lymph nodes too. Okay? So a lymph node is an encapsulated structure, and it has different zones, right? So you have T cell zones and B cell zones. And the cells generally stay in separate regions within the lymph node. But remember, I talked a lot about immunosurveillance and showed a couple of videos. These guys are never just sitting there. They're always moving, and they always need to move because they need to encounter their appropriate antigen so that they can become activated. So they'll traffic between lymph nodes, they'll traffic into circulation, they'll traffic into the spleen, and they'll just keep doing that for their lifetime until they encounter antigen or undergo apoptosis because they've been hanging around too long. Okay? So you have T cell and B cell zones, right? And that dendritic cell will bring that pathogen into the lymph node, to the lymphatics. And what it'll do is it'll internalize it and degrade it, and it'll present antigen. Okay? So when you do that, right, an antigen presenting cell, professional presenting cell, has what type of MHC molecules on the surface? Class 2, right? So B cells, macrophages, and dendritic cells are professional antigen-presenting cells, and they have class 2, which is not found on other cell types. Okay? They also have class 1, but everything has class 1, but they also have class 2. Okay? So they present antigens through class 2. So they're going to degrade the antigen into little peptides. And for this type of antigen generation, something for like a macrophage, for example, that would do that, or a B cell. What mechanism, what pathway for MHC loading is occurring here, right? There, Dr. Kalan talked about two different pathways. There's the endogenous and exogenous pathways, right? So endogenous is for pathogens that are already inside the cell. So things like viruses that have managed to get inside a cell, they're going to be degraded through the endogenous pathway. Pathogens that are brought into the cell to be degraded are going to come through the exogenous pathway. So through the exogenous pathway, those pathogens are going to go through the lysosomal compartment. And be degraded into small peptides and loaded into class 2 molecules. Right? So remember, he was talking about class 2 molecules. Class 2 molecules have to have something in their binding site. So until they encounter their peptide, they're going to have what in that binding site? The invariant chain, close. Invariant chain. Right? So that sits in there, protects that site, from, and also protects the molecule from being degraded in the lysosomal compartment. And there's all kinds of enzymes that are designed to chew up pathogens, so you don't want it to chew up the MHC molecules. So that invariant chain will be degraded, peptides will be loaded, and that MHC2 molecule with its peptide will go to the surface of the cell, where you'll have class 2 expressed on the surface of, let's say, dendritic cell. So you have class 2 molecule, peptide, and then this complex of peptide and MHC2 molecule is going to be recognized by the T cell receptor. Okay? For that particular antigen, right? So the T cell receptor sees the complex, it doesn't just see the peptide, it sees the entire structure. And for activated T cells, that's the first step. And we'll talk a little bit about that yesterday. So the interaction of the TCR with the MHC complex, and that's also facilitated by CD4, which will bind to the MHC molecules. So that's the first step in activation of the T cell. Okay? So that's going to send a signal into the T cell. Something's happening here. That whole structure is held together by adhesion molecules that come into play that help keep that interaction for a longer period of time. The next step that has to happen to activate that T cell is going to be, anybody? Okay, co-stimulation. You have to have co-stimulation. If this interaction doesn't get co-stimulated, the T cell will become allergic. It won't do anything. It won't respond. Eventually not. Okay. So co-stimulation, CD28, and the B7 molecules. So that's the second step. And when that happens, then you're going to start activating T cells enough that they're going to start producing their own cytokines. So for T cell activation, one of the key cytokines for activating them, IL-2. And the signals that the T cell receives can also come from cytokines produced by dendritic cells too. So they can produce cytokines and shape that T cell response. So now you've activated T cell, it's going to undergo proliferation, differentiation, and depending on the signals it's gotten, it's going to become either a Th1 type cytokine, which produces cytokines that are involved in cell-mediated immunity, or a Th2 type cytokine, which is involved in humoral immunity, helping to activate B cells so they'll produce antibodies. Okay? So that's critical. So we have to get that B cell activated too. So there are a couple of different ways B cells can get activated. And erase some of this. Here's our B cell. And B cells have on their surface immunoglobulin. And a naive B cell has IgM on its surface. Right? So if the antigen is really large and it has a lot of carbohydrates on it, for example, you can get cross-linking of that IgM molecule on the surface of the B cell. And that's enough to send a really potent signal into the B cell to help activate it. So that's T-independent activation. You don't have to have T cells to help activate B cells through that mechanism. Okay? So that's one mechanism. And then there's also T-dependent. And that's when T cells are going to directly interact with this B cell. Okay. So B cells, remember, are antigen-presenting cells. So they can take that antigen inside and degrade it and put it up on the surface, just like the dendritic cell did. So we'll have the peptide there and the TCR, right? So that's going to be one of the signals. You're going to get co-stimulation. Again, you have to have that. So that's the first step, that interaction of the MHC TCR complex. And then you're going to need co-stimulation. And so here we're talking CD40 ligand. That's the co-stimulation step. That's the second step. And then these guys are going to start producing cytokines, and that's the third step. And then you're going to get this B cell activated, and it's going to undergo differentiation with the ultimate goal of becoming a plasma cell, which no longer has antibody on the surface, but basically becomes an unbelievable antibody factory. Just turns out tons of antibodies, thousands of molecules per second. It's just amazing how much it can do. And then you'll also get memory cells from that process. So you'll have T memory cells, and you'll have T memory cells from that as well. So that activates the adaptive immune response. So we're at time. Anybody have questions? So what I have, if anybody wants to stick that for a couple of minutes, is some sample questions. We can just run through just a few of them, just so you can kind of get an idea of what it is that you're going to see. And again, I want to stress that if you know the study guide points,
that ain't gonna work. Oh, it's gonna fall back this way. Oh, got it.
Okay, so these are three examples, some things that might be on the test. So things like, what's humoral immunity? Is it heightened immune responses due to laughing? Probably not. Physical barriers such as skin, MHC molecules, immunity mediated by antibodies and B cells. That looks pretty good. And innate immune responses mediated by T cells. Well, T cells don't do innate immunity really, right? So it's got to be antibodies and B cells. So what's innate immunity? So what kind of responses there? Toll-like receptors to target lots of pathogens. Phagocytic cells to eliminate pathogens. Doesn't really generate memory. Non-specific responses for self versus non-self. All of those sound pretty good for innate immunity, right? So the answer would be E. All of the above are correct. What's the primary lymphoid organ? Well, that's pretty obvious, I think. Right? Spleen, thyroid patches, lymph nodes, all are secondary lymphoid organs. Only the thymus and the bone marrow are primary. How could you determine what specific class of immunoglobulin is present within serum or mucosal samples? Just by molecular weight? Probably not. Some of them are pretty similar. The hinge region? A lot of variability there. Whether you digested them with pepsin or cocaine to give you C fragments or FAD fragments? Not really. Some of them would look alike. What if you look at the amino acid sequence of the heavy chain? Then you could tell whether it was a gamma chain, an alpha chain, a mu chain, an epsilon chain. You can tell what you think. So if you know the sequence, you know what that is. Okay. What makes something immunogenic? Right? Size, smaller or immunogenic? Pulse. Chemical composition, proteins more immunogenic than lipids. Foreignness from the species immunized. All the answers are correct. Only B and C. Looks like E to me. Okay. Isotype switching. The light chain goes from capital N. Which chain's involved in isotype switching? It's not the light chain.
It's the heavy chain.
It's the heavy chain. Right? So the heavy chain that was originally giving you an IgM antibody can become part of an IgG antibody, or that maybe you start making two at the same time. All the answers are correct. So it's just got to be this thing here. B is correct. So cytokines, for example, so cytokine biology or activity is regulated by transient secretion. We'll talk a little bit about that. Receptor expression. If the cell doesn't have a receptor for a cytokine, it can't respond to it. The effects of antagonistic cytokines, well, we know that occurs. So all the above answers are correct, or none of the answers are correct. I think it's going to be D. A little bit on complement. C5b fragment generated on complement activation doesn't play any role at all in the immune response. Combined covalently to surfaces. No, doesn't have a bioester bond, can't do that. Interacts with activators of the complement system by ionic interaction. Starts the formation of the membrane attack complex. I think that's the answer there. Leukocyte firm adhesion occurs in arteries and arterioles. Postcapillary venules, high venules, very specialized structures which have different repertoires of adhesion molecules there, so cells will target appropriate tissues when they need to. Requires selectins. Firm adhesion, I don't think that was a selective mediated process, right? That's rolling. Right? So that can't be it. Requires integrins and CAM molecules like ICAM1. That looks kind of good. That's firm adhesion. Uses lots of different families like PCAM and JAMs and integrins and stuff. That's the transmigration stuff. Yeah. So if you know the different steps and the different families of molecules associated with that step, that's all there is. Right? Stimulation of T cells through the T cell receptor. In the absence of a co-stimulatory signal through CD28, you get superactivation of the T cell. You get lots of IL-2 production, inactivation of the T cell, energy. That's what happens, you get energy. So you don't get differentiation and you don't get memory cells because without that co-stimulation step, that's it. So if you know the 3 different steps, you know the T cell receptor and the MAC complex, and the co-stimulatory molecules, the only other thing to throw into the equation is cytokines, that's T cells. B cells basically kind of the same thing, except the antigen receptor is the antibody, and co-stimulatory molecules are a little bit different. The cytokines, that's all there is to it. So frequent viral infections could be due to a deficiency of mast cells. I don't think so. Do you think of mast cells as the key players in controlling viral infections? CD8 cells? Those are referred to the cell type aside from NK cells that are important to control them. So these other cell types not involved. So that's kind of what the questions are going to be like. So you can get highlights